Hello everyone, welcome to Ash Jottings. In today's video session, let's talk about the different forms of DNA. So let's get started. Types of DNA. So let's just have an introduction. So there are 6 different morphological forms of DNA double helix which have been described. These are, as you all know, A, B, C, D, E, and Z. But out of this, only the B and the Z forms of DNA occur in cellular DNA. Cellular DNA. So these conformations occur as a cellular DNA. Others are mostly seen under laboratory or experimental conditions. These are seen in the experimental conditions. So all DNA molecules of any source, let it be natural or artificial, can take up the two forms, that is A and B forms. Okay, now some DNA forms are interconvertible. Interconvertible. And the difference between all this DNA lies in associated with the, we just write it like number of base pair present in each turn of DNA helix per helix, or each turn of. And second one is the pitch or the angle between each base pair. And third one is the helical diameter of DNA molecule. And the last one lies the right or left handed or handedness of coiling of double helix of double helix. So there are different forms of DNA which are present, that is A, B, C, D, E, Z. And out of this, you can find only two types in the cellular DNA, that is the B and the Z, and rest are found under laboratory or experimental conditions. So the few DNA forms are interconvertible, and number of base pair per, so how they actually differ depends upon the number of base pair per helix, the pitch or the angle between each base pair, and the helical diameter of the DNA molecule, and the right or left handedness of coiling of double helix. Now let's just look into each of these forms one by one. Now let's start with the B form of DNA. Just move the- So you have the B form of DNA. So when you talk about the B form of the DNA, this form of the DNA occurs in all living beings, occurs in all living beings under normal condition, that is, they are naturally occurring form. They are fully hydrated form. So that's why they are also called as wet DNA. It occurs in low concentration of salt or low salt concentration, but it has high degree of hydration. So we already learned that is it fully hydrated. Say around 92% of relative humidity. Okay. Now, the beta helix is thinner and taller. The beta helix is thinner and taller with it has a wide major groove and narrow minor groove. Okay. It so as of now, you know that these are the B form is the common form which we are studying right from the beginning, right? So I think all these points you must be aware of it very well. It's each coil or turn. It's each coil or turn measures 34 angstrom, right? Or you can say it as 3.4 nm. Now each turn has 10 base pair, and each base pair occupies 3.4 angstrom. That is, you can just divide 34 by 3.4 will give you the number of base pair per turn. Okay, it lies same in all the forms of DNA. Now, the two strands are polypeptide. We know that it is two strands are nucleotide chains wounded on right-handed coils, and each strand follows a clockwise path. So who discovered it? We already know it is Rosalind Franklin, who actually described the form, and later on it was Watson and Crick who describe the double helical structure. So we have seen all these things. So basically your DNA will be like this. Okay, very bad drawing. Okay, when you see the helix, you have the axis to run parallel to this, and you have the bases, right? So I think we have learned about this, so I'm not going to talk more about this. Okay, now let's move on to the next form, that is A form. Let me just write it here. Okay, A form. A form is also right-handed DNA. Okay, it is dehydrated and more compact than more compact form of DNA. It actually occurs under decreased hydration, and it needs increased concentration of Na plus ions. It is also polynucleotide chains coiled around clockwise, okay, clockwise, and as I told you, is having a right-handed configuration. Now this A form of DNA is more compact, it is compact and also bulky. Its helix are short. Helix is short and wide with narrow and deep major groove, and it has a wide plus shallow minor groove. So it does have both kind of grooves, which is seen in form B of DNA. It has 11 base per turn. Okay, and the diameter is diameter is 23 Angstrom, and the distance between the base pair is distance between the base pair distance between the base pair is 2.7 Angstrom instead of 3.4 in B form of DNA. Now the orientation of the bases is also different, that it is will be slightly tilted and displaced laterally in relation to the axis of the helix. And a helical form is adopted by RNA or RNA DNA and also RNA helix. And this A DNA is actually assumed that they are derived from bDNA. Okay, so let me just give you a rough sketch of how it looks. So here the bases, nucleotides will be slightly tilted and displaced. So it will be, instead of, you see, in the B form, it will be like this, right? So here it will be slightly displaced, maybe here, here, okay, might occur slightly displaced. That is why it is tilted and displaced laterally. Okay, now this is all about the B form of DNA and A form of DNA. Now let's talk about the D and E form. D and E form of DNA. So these two forms of the DNA are also right-handed. Okay, right-handed, and they have a slight different conformations. And these two occur in helix, which lacks occurs in helix, which lack guanine. That is, which means actually they have alternating A to T base pairs. Okay, they have alternating A T base pairs, and these have 8 to 7 nucleotides per turn respectively, and they are seen in special environment and do not occur in vivo. No in vivo. So this DNA has been detected in nature only in T2 bacteriophage. Bacteriophage, yes. So this is all about D and E form of DNA. Okay, now let me just give you a rough idea how the base are aligned here. See, can just see. Um, oh, cannot well. So here it is just slightly, as I, and the B, it will be just like, you just have the A and T bases, right? So it will be like this. Sometimes it just goes here. Okay. The bases will be arranged in this manner. Okay. So now let's talk about the C form of DNA. C form of DNA, which is also again a right-handed DNA. It actually occurs even at a greater, just like the form A, greater dehydration, which is usually not found in the cells or under normal conditions. Yes, so it has around 9.3 base pairs per turn of helix instead of 10. The base pair are tilted relatively to the axis. So here the base pairs will be tilted relative to the axis of helix. helix. Okay. The helix is only 19 Armstrong in diameter, and it has, so it is, because it has just 19 Armstrong, so it should be narrow and less compact than, you can say, A DNA and B DNA. So it occurs in a relative humidity around 66 percentage, and also a medium which contains lithium ions. Let me just draw the picture here. See here, the bases will be tilted relatively to the axis of the helix, so it will be present on the inside, but in a like this, slightly they will be tilted. Okay, now the last form, let me take a new page, is about the Z form of DNA. Z form of DNA. Now this is only the left-handed DNA, right? The rest of the things are all right-handed. It occurs in small proportion or small quantity or proportion in cellular DNA. Okay, small amount in cellular DNA. It is called zDNA because the phosphodiester backbone of its polynucleotide strand passing through the phosphate group, and it follows a zigzag course. That's why it is called zDNA. That is, the phosphodiester backbone of its polynucleotide strand, it passes through the phosphate group. So it gives it an appearance of a zigzag. So Z stands for zigzag. Okay, zigzag. Now, this was actually discovered by Alexander Ridge around in the 1979. It is the skinniest DNA with a base pair of 12 base pair per turn. And one helix, one complete helix, you can say, one complete helix is 45 Armstrong against 334 in bDNA, right? The diameter of this DNA is 18 Armstrong, which is against 20 Armstrong in bDNA. It has only one groove present. So that is major groove which is present. Oh, sorry, it should be minor, I guess. It should be minor groove, and major is absent in the Z form. It is less stable. It is less stable than bDNA because, you know what, the phosphate groups which are present in the polynucleotides has negative charge, and these phosphate groups, they lie much closer. Okay, so that will cause a repulsion which actually leads the Z form of the DNA to B form of the DNA. Okay, it is less stable because of the phosphate group, which are very close to each other. Unlike here, they are close, but as they actually go and bind to certain other cations, so it will avoid the repulsion in the case of B form. But in the Z forms, they are pretty closer, and that leads to the repulsion, which actually converts ZDNA into ZDNA to a bDNA. It is formed. Okay, now next point is, it is formed when purine and pyrimidines are present. Okay, purine plus pyrimidine when are present alternatively in the chain. Okay, that is GC or it either should be present alternatively. Very sorry with my handwriting. Now, its repeating units are dinucleotide against a mononucleotide. So, in the bDNA, it is mono. Here, it is the dinucleotide. Let me just write it like this. Dinucleotide repeating units. It is more compact. And also the zDNA sugars have sugar residues. Sugar residues have alternating or alternating orientations. It can be present in high salt concentration, or you can even say special cation. It explains the repulsion. So what happens when the bDNA is brominated, or it is methylated? It changes to zDNA. Okay, you can say when you actually stretch the bDNA, it will change to zDNA, and if you compress, it is going to change to A form of the DNA. So that's all about the types of DNA. Let me just quickly, let's get back what we have learned. So initially we thought introduction, where you know the different types of DNAs which are present, and what actually how they differ. The four criterias have been mentioned, that is the number of base pair per helix, and the pitch or the angle between each base pair, and the helical diameter of the DNA molecule, and the right or left handedness of coiling of double helix. So next we, okay, we spoke about B form of the DNA, which is a double helix DNA, which is a common form. And I don't have to again brief all those things. Only thing you remember, it is also called as wet DNA because it is fully hydrated form. Now the A forms we have seen, and then we saw the D and E, C form, and finally the Z form. Okay, thank you.